Hi. Um, so I'm well aware that I uploaded a video of me crying, and you may have seen it. And if you haven't seen it, I took it down, so don't even ask me where you can find it. But might be like, why? Um, I was crying about a lot of misfortunes I have in my life. One of which being the lab final I have today at 10, like in 8 hours. And I finally decided to sit down, force myself to look at all the material and work, put myself to work, you know? And so I finished all the notes. So now this is gonna be the final lab review stream, whatever, before I try hopefully to sleep and ace the exam, hopefully. So I have me here, girl dinner, and so it consists of carrot cake and mac and cheese. I made the mac and cheese myself. And it wasn't like Velveeta or anything. I made it from like scratch. Except for I have um like pasta, but I didn't make that from scratch. But anyway, I also have a sandwich with, oh my gosh. I have a sandwich with my garlic butter, like garlic confit butter or whatever. You're like, how Nini, how are you gonna have the time to sleep if you're gonna eat and then talk about this? I don't have the time, but I do have time. And I put that in the sandwich, and I'm about to eat it up right now. And then top it all off, I have me a tall mug of delicious milk. When I'm finished with the milk, I have cherry Coke. And you're like, nee, this is not healthy. Well, I'm not in nutrition heavy classes yet, so it doesn't matter. Until I am in nutrition, I can eat whatever I want. And it's not like tonight's a normal night. I went workout. I went to work out. I ran, so I feel like I get a little treat. I get a little sweet treat today. Anyways, I just finished with my cake, and I'm gonna eat the mac and cheese. It's so hot. The mac and cheese is too hot for me. I microwaved it for like 5 minutes. Look, I made it with gnocchi. I think that's how you say it, but it's so good. Uh-uh. Alright, I'm gonna give this to like 5 minutes, and then I'll start reviewing. Let's say 6 minutes. I'm still trying to eat. Honestly, at this rate, let's just go 6 minutes. Or 7. Or 10. Here, let's say 10. Let me eat for 10 minutes. And then I'll start reviewing. Eating. So tomorrow when I wake up, I just go to lab and I don't have to eat. And I'm not gonna sleep too much. Probably just gonna sleep for like 2 hours. And then I'm gonna go take the test, ace it, go home, sleep. Look, it's so hot, it's fogging up my glasses. All right. It's when I say all right, I mean like I'm about to eat this sandwich. I'm not about to start the stream. Well, the faster we finish this, the faster I can go to sleep. But I'm not finished eating. Wait! What is the timestamp doing? It's 10 minutes already? Girl. Okay, give me a second. I don't know. Cheese just fixes everything. I've been eating so much cheese lately. They call me the dairy fairy with how much I be cheesing. It's like falling apart. Okay, 15 then. 15 minutes. Sorry, I'm done. We're done. I just choked on some milk. That's not very dairy fairy of me. Anyways, I didn't expect that for me to take that long. I didn't expect for me to eat slower than it took to build the Roman Empire. Like that was just way too unnecessary. So, anyways, it's actually 15 minutes. We'll start talking about this. I will start talking about where we left off, basically. I will talk about labs 8 through 15 pretty much. And then I will try to go over the first few labs. And I can't find my thing, so where's my I left a paper. So first I'm going to talk about the labs of the second half of the semester, and then I'm going to talk about the labs of the first half a little bit, like in no review. Anyways, So this half of the lab is talking about animals. So there's 3 phylogenetic groups. There's Porifera, which is like a phylum, and then Radiata, and then Bilateria. So Radiata has 2 phylums or phyla, which is Cnidophora and Cnidaria. I don't know. I'm gonna say Cnidaria. I don't know, bro, but you know what I'm talking about. Anyways, okay, so Porifera are sponges. So there's no tissues and stuff. So they have spicules and spongin, which are made of collagen. The smaller ones are the spicules, and then the longer branch ones are spongin fiber. So the water flow goes from the pores to the spongocoel, and then to the osculum, which is the opening. And the water flow is aided by choanocytes, which are collar cells. So there's 3 body types. There's simple, which is also called asconoid, intermediate synconoid, and complex leuconoid. And by the way, the spongocoel is the middle of each of these bodies. So we're talking about Cnidaria. That's jellyfish, like just jellies in general. So they have radial symmetry, they have tissues, and they have tentacles, stinging cells, which are called cnidocytes, and their opening is a mouth and an anus. So they have either a polyp or medusa or both lifestyles or stages after larval stage. There's 3 classes. There's hydrozoa, which are hydroids, and there's 2 examples of these. There's Obelia, and then also the Portuguese man of war. There's Scyphozoa, which is jellyfish, and then Anthozoa, which are sea anemones and corals, and these are only polyps. They have sea anemones are muscular and sessile, but all of them. And corals have calcium carbonate exoskeletons. Okay, so we're gonna move on to the third group, which is Bilateria. These animals have bilateral symmetry and associated with cephalization, which is having a true head. And so yeah, anterior, posterior, front, back, dorsal, ventral, top, and bottom. So they could either be acoelomate, pseudocoelomate, and colmate. So acoelomate means like there's no body cavity. Pseudocoelomate means there's the mesoderm on one side and then the endoderm on the other side, so it's like half and half. And then collimate means there's a true body cavity surrounded by like the mesoderm on every side, like on both sides. Okay, so Bilateria has pretty much 2 clades, like major clades. There's protostomia and then deuterostomia, and they were kind of differentiated by what evolves first. So protostomia, the head evolves first, and then the deuterostomia means the anus. We're like the anus evolves first. So there's molting protostomia and non-molting protostomia. Non-molting protostomia are called lophotrochozoans, and then molting protostomia are called ecdyozoans. So we're first going to talk about lophotrochozoans, which is really long, but I don't know. We have 3 classes 3 phyla and about like 10 classes. So first one's about Platyhelminthes. Platyhelminthes are flatworms. They are mostly parasitic, hermaphroditic, and they have dorsal flattening. There's a Trematoa class, which are flukes. Cestota, which are tapeworms, and Turbularia, which are planaria, and these are they kind of look like a slug, but they're not really. They're flat. And tapeworms kind of look segmented, but they're not really. Okay, so we're gonna move on to Annelida phylum. These are segmented worms. They have setae. A lot of them do. So there's a class Polychaeta, which are clam worms. They look very scary. They look like a worm with like a whole bunch of spikes. And then we have Ogliochaeta, which are earthworms. They have clitellum for reproductive pores close to their head. And then we have Hiradinia, which are leeches. So they don't have setae or parapodia, but they do have suckers. And then last but not least, we have the phylum Mollusca, which are mollusks. They don't molt, and they're unsegmented. The class is Polyplacophora, which means many plates. These are chitons. They're like they look like oval, and they look like a turtle, pretty much. It's a large foot and 8 valves. The gastropoda class is means stomach fruit, and these are snail, slugs, and nudibranches. The class Bivalvia are scallops, oysters, and clams. Class Cephalopoda are octopus, squid, cuttlefish, etc. So yeah, we are done with that. Okay, we're going to talk about molting protostomia now. These are ecdysozoans because ecdysis is molting. It's a fancy word for molting. So, first we're going to talk about Nematoda, and then we'll talk about Arthropoda. And so, those are the two phylum, but Arthropoda has like a whole bunch of subphyla and then classes. So first we're going to talk about the easier one, Nematoda. These are roundworms. They are pseudocollimates and dioecious, and the example is Ascaris, and the female is much larger and thicker than the male one. And then arthropods, which means joint foot. These are segmented with fusion and cephalization, and they have they're collimate, and they have a chitin exoskeleton. So there's 4 subphyla and then 6 classes. So we're going to talk about subphyla Chelicerformes. These are paired Celicerae, paired polypaps, 4 legs. There's a class Meristomata, which are horseshoe crabs, and then Arachnida, which are spiders, scorpions, mites, ticks. We have subphylum Myriapoda. There's a head, segmented trunk, unbranched appendages. The class Diplopoda is millipede, and it's called diplopod because there's 2 pairs of legs per segment. And the class Chilopoda is a centipede, which means just has 1 pair of legs per segment. Okay, subphyla Crustacea, and these are all under arthropoda, by the way. They have a head and 2 pairs of antenna and mandibles. So there's a class Crustacea. Oh yeah, so like Crustacea is the subphylum and the class. So these include barnacles, crawfish, crabs, lobsters, shrimp, all that stuff. And then subphylum Hexapoda. This includes the class Insecta. So insects, bees are an example of that. Okay. So that's it for the protostomes, sorry. Okay, now we're going to talk about deuterostomes. There's 2 phyla. There's Echinodermata and Chordata. So, first, Echinodermata has unsegmented collimates. They have a water vascular system, dermal endoskeleton with spines. So they're kind of like spiky-ish. And so the class Asteroidea, these are sea stars, like starfish. There's central disc and then arms. And then Ophiuroidea, these are brittle stars. So these are a central disc with like legs too, but they're longer and they look like a snake. That's why you can remember by thinking like Ophis meaning snake. The Echinodea class, these are sea urchins and sand dollars. And Holothuroidea is sea cucumbers. Okay. Now we're going to talk about the second subphylum of deuterostomes. So chordata, these are they have a notochord, a gill slits, postnatal tails. So 3 subphyla, 7 classes. They're subphyla Urochordata. These are tunicates and sea squirts. They are enclosed in a cellulose tunic enclosure. Subphylum Cephalochordata has lancelet. It's a lancelet and amphioxus, and these kind of like look like, I don't know. They look like lancelets. Like they look like spears, but like swimming around. Subphylum Vertebrata. These are vertebrates. There's a class Agnatha, which are jawless fish. Example: brook lamprey, and then the class Chondrochites. Chondrochites are cartilage fish. So, example: sharks and rays. Class Osteichthyes are fish with bony exoskeletons. So, example is a yellow perch. The class Amphibia, these have smooth no-scale skin and 4 legs. So, example: frog, salamander. Class Reptilia have scaly skin and 4 legs. So, example: lizard, turtle, and snake. Class Aves are birds. Class Mammalia are 2 pairs. They have 2 pairs of legs, hair, and mammary glands. So, us, pretty much. A little bit about just the anatomy of stuff. So okay, so we're gonna talk about blood cells. Red blood cells are the most prominent in the blood smear, so you'll see those everywhere. The platelets are kind of like small fragments. Neutrophils have a lobed nucleus, so they're just kind of like big cells with like you see the other little cells inside of it. Cell-looking structure. That's just structures inside of it. Lymphocytes have a large nucleus and small cytoplasm, so you'll see it. And then how basically the anatomy of the lower part of the body goes is you got your kidneys, and then there's 2 tubes called ureters lead down to the bladder, right? And then kind of like above your kidneys are your adrenal glands. Okay, so for women, right kind of like where the bladder is, there's the uterine horn, and then oviduct on like okay, so there's a uterine horn, right? And it leads to oviducts, and then it leads to your ovaries. And then down here, you got your bladder going down against your urethra, then your vagina, and then the anus is behind, kind of like it's posterior, I guess. And then, adrenal and kidney. And then for males, in the middle, there's the bladder, but to the sides, you have the seminal vesicles. And then underneath the seminal vesicles and the bladder, you'll see your prostate gland. And then to the side, you'll have little tubes called vas deferens. And then the testes with the epididymis connecting the testes and vas deferens. And then the scrotal sac helps encase the testes. Anyways. And the other part is the penis with the urogenital orifice. Okay, so that's reprod and excretory. Now I want to talk about heart. Actually, let's talk about heart after. Actually, let's talk about heart now, okay? So, the heart, when you're looking at it, the right side is like when you see it on your paper, the left side is right, and then the left side is the left side is right and then the right side is left, basically. So the top, the top part is the atrium. So it goes right atrium, left atrium, and then the lower ones are the ventricles. The tube on the left side that you see, or on the right side of the body, but the left side of the chart, that's the vena cava, and this carries like blood from the body into the heart, and then pumps it back out to the lungs. So from the body to the heart to the lungs. And then the structure on the right or the left side of your body is called the aorta. And this brings blood from the lungs through the pulmonary arteries or pulmonary veins, my bad. And then pumps it out to the upper and lower body. So this is the path of blood, all right? So the blood from your upper and lower body, it comes into the heart from the sorry. It comes into the heart through the superior and inferior vena cava, goes to the right atrium through the left ventricle to the pulmonary arteries, pumps out to the lungs, comes back into the heart through the pulmonary veins, goes through the left atrium, left ventricle, and then through the aorta, through the aortic arch, and then moves back to the upper and lower body. That's how it goes. All right, that's that. This you can review it. You can review it. I'm not gonna talk about it. I'm not. I really am not. Okay. Little thing. Talk about. Euglenozoa are flagellates. Example: trypanosomes, gymnoamoeba, the amoebas. Example: amoeba. Ciliophora, ciliate. Example: paramecium. Apicomplexa, example: plasmodium, causes malaria. Foraminifera, these are marines, have spiral calcareous tests, so they're strong. And then radiolaria are marines, they have siliceous tests, and they are fragile. Okay, those are protozoans. And then we're going to talk about algae now. So chlorophyta are green algae. Examples are Chlamydomonas, Gonium volvox, Hydrodictyon, Odigonium, and Ulva. We have Phaeophyta, which is brown algae. We have Rhodophyta, which is red algae. And we have Bacillophyta, which are diatoms. We don't really talk about them. Okay, the third part of the protist are slime molds. Myxogastria are plasmodial slime molds. They have sporangia, and example is Fusarium. Okay, so now we're going to talk about plants. We have non-vascular plants first. So 3 phyla. Bryophyta, which are true mosses. They have a foot seta and capsule. Hepatophyta, which are liverworts, and they have gemmacups. And then Anthocerophyta, which are hornworts, and they look like green beans. Seedless vascular plants. They have a xylem for water and minerals. They flow up only, and they phloem for carbohydrates and photosynthase, and they flow up or down according to needs. Lycophyta is a phylum. These are club mosses. They have a strobilus. They kind of look like, I guess, I think they look like, I don't know. I don't know. Just hope that you don't get that. Anyways, Psilophyta, they have no true leaves. They're dichotomous. They have yellow bumps, so they look like STD leaves, I guess. And I think Lycophyta is like that plant that you eat. I forgot. Anyways, Arthrophyta are horsetails. They have hygroscopic elaters. Oh, that's really cool. I remember talking about that. Pterophyta are ferns, and they have sori and a prothallus. Okay, and then the third part, so those are seedless vascular. Now we're going to talk about seed plants. So we have gymnosperms and angiosperms. So first, the gymnosperms, we have the phylum Cicadophyta. They look like a palm tree and fern club. Ginkgophyta, you know what that looks like. Gnetophyta, they are source of, the example is ephedra, source of ephedrine, looks like small spiky grass, like small dried spiky grass. Coniferophyta is conifers, have pine trees, and they have staminate pollen cones. Pollen cones are bigger than staminate. And then they also have micropyle, which is the opening of the pollen tubes. Okay, now we're going to move on to angiosperms, which are not naked seed sperms, but they are protected seeds. So, of course, they have flowers and fruit. So, there's the only phylum Anthophyta. So, we have monocots and eudicots. So, just how many leaves? Seed leaves. Okay, so there's 4 whorls that attach flower parts to the receptacle. So the calyx and the corolla are sterile. So the calyx are the sepals, and then the corolla are the petals. And then we have the androecium, which is the stamen, which is male and fertile. And then the gynoecium, which is the carpel or pistil, and it's female and fertile. We have complex, incomplete, perfect, imperfect flower structures. We have contation, which is fusing within a whorl, and then adnation, which is fusing between whorls. So, example, sepals and petals fused. There's radial symmetry, bilateral symmetry. Radial is like you can draw a line anywhere, bilateral means you can't. There's the exocarp, mesocarp, and endocarp of a fruit. Fruits can be simple, aggregate, or multiple. Can be fleshy or dry. Okay, so simple is derived from one pistil, aggregate is separate carpels, and then multiple is multiple flowers in one like thing, I guess. So, okay, the male parts, again, to review, the known collectively as an androecium, they have individual stamens. Each stamen has an anther and pollen sacs. Female parts is the gynoecium, and they have individual carpels fused to make a pistil, and also a stigma, a style with ovary and ovules. Okay. And then, in the mature embryo sac, there are 3 antipodal cells in the end, away from the micropyle. I don't know what that means. Okay, so I'm talking about growth of a seed now. The radicle is the first organ to penetrate seed coat. Then you got the primary root, and then the root hairs, then lateral roots, the hypocotyl. Then it'll turn into a cotyledon, either like 1 or 2 seed leaves. So after the hypocotyl straighten out, the epicotyl with primary leaves forms, and then the terminal bud is visible. And then one main stem has nodes and internodes, which is the attachment area of the stems and stuff, and the area in between. Not the stem, but the leaves. There's 3 types of leaves on the main stem. There's the cotyledon, which is probably withered by now because it's old. The plant is old. And the primary, which are above the cotyledons, they have they're simple leaves, and they have petioles, which are just like stalk. And then compounds are all leaves beyond first node. So like, yeah, you'll at first node it'll be like opposite leaf arrangement, and then alternate in the nodes beyond. So the axillated bud is above the point of leaf attachment for each node. And then leaf abscission means they drop from trees after an abscission forms from a petiole base. It just happens so like as leaves get older, there's less auxin hormone, so there's more leaves that fall, I guess. Apical dominance is basically when the terminal bud at the top of the, or like the apical meristem, it prevents development of the axillated buds. So yeah, a little bit about dwarfism. So gibberellins are a group of plant hormones. They control plant processes like growth and elongation. So many plant dwarf mutants, they don't respond to auxins and cytokinins, which are like hormones that like promote cell growth and stuff. They grow when gibberellic acid are applied. So like if auxins don't work, if you don't add auxin in, or like if you add auxin or cytokinins and they don't work, gibberellic acid will help grow the plant. So yeah. Etiolation is the growth of plants in the dark. It increases stem elongation and like helps with like starvation because it would need less light. But once the plant is reintroduced to more light, it will eventually like raise its standards for how much light it needs. And then we got fungi, which are eukaryotic. They have cut cell walls. They reproduce asexually or sexually. They can be unicellular, multicellular, or a mass of hyphae. They have condidophores, and they have zygotic meiosis. I guess to review the sex cycles, so animals have gametic meiosis, and then fungi have zygotic, and plants have alternation of generations. All right. I think I reviewed everything. It's been 40 minutes. I want to see the mitosis chart so I can talk about it. Like, oh, you also have to look through the dissection label stuff tomorrow morning because I don't know anything about that. All right, so life cycle study guide. Let's see what's happening here. That doesn't help me. That doesn't help me, thanks. That doesn't help. Well, I don't know. Let's pretend to go to lab 4. No, let's do lab 3. Labs 2 and 3, PowerPoint, and then review the life cycle. Sorry. Okay. So in sporic meiosis, it starts off as either... I'm not gonna even explain that. I'm not gonna lie. I don't know what that means. I'm not gonna explain that. I don't know what that means. That's actually crazy. Yeah, we're not gonna go over that, but we are going to go over lab 1. Okay, where is the PowerPoint? I'm like almost done. Okay, let's see. Let's see, where am I? Okay, so usually the gametes start as one end, then they get fertilized, then they become 2 end zygote, and it goes through mitosis, then it's still 2 end, and then it goes through meiosis, and then it becomes 1 end, then mitosis again, and then 1 end, I guess. I don't know. Okay, so zygotic meiosis. Zygotic undergoes meiosis. So basically, gametes start as 1 end, and then they get fertilized, it becomes 2 end, the zygote. The zygote goes to meiosis, becomes 1 end, and then goes to mitosis, and then becomes 1 end. So like it lives as haploid basically. And then through gametic meiosis means gametes form directly by meiosis. So let's see. Okay, so so basically zygotic meiosis, the zygote, so basically mitosis happens immediately after fertilization. So most of the life is haploid. The gametic meiosis, it happens... No. No. Zygotic meiosis, the mitosis happens after meiosis, like right before fertilization. So it lives as haploid. And then gametic meiosis, it happens right after fertilization, which means it lives as diploid. Then undergoes meiosis, and then 1 end, and then fertilization again. Alternation of generations, there can be mitosis after the zygote or before the gamete, so that's how it is. And yeah, that's pretty much it. I think I have reviewed everything. I am now going to go to sleep. Finally, hopefully. Let's try to... It's 3:21. Let's try to shoot for 3:45. If I sleep at 3:45, I can probably get up at... well, let's go to sleep. All right. So if I sleep at 3:45, and I wake up around 8 to get ready. Well, let's wake up at 8:40. That would give me about 5 hours. Okay, but let's wake up at 8 so we can review our other notes. So I will get 4 hours and 15 minutes, and then I can get other 4 hours um after I get back home from the lab. So yes, yay. Okay, thanks for spending your time reviewing with me, and I'm gonna go to sleep now. Bye.